Hello, and thank you for coming to my talk today, our Monarchs Pollinators. I am Monroe Pruett, and my mentors are Dr. Burke and Dr. Mackenzie Taylor here at the Department of Biology at Creighton. So here is my research poster. Feel free and pause here to get some background details, but I will be going over everything in the next few slides. So to introduce my topic, my big research question is, are monarch butterflies successful pollinators? And to define a successful pollinator is a mechanism that carries pollen from one flower to another of the same species and then deposits the pollen that will germinate the flower. And so I was also looking at a couple questions of, if monarchs are successful pollinators, then how do they carry pollen? Where do they carry it on their body? And are they depositing the pollen to the flower of the same species? And a little sneak peek here, I have found that monarchs can pick up and transport pollen. So it does kind of cover those two areas of they're able to pick up pollen from a flower and transport it. We just don't know if they are able to deposit the pollen onto a flower of the same species and if that pollen ends up germinating the flower. So kind of going into my methods for this study, my photo here. So I did my study at the Glacier Creek Prairie Preserve in the summer of 2020. Here is a photo of the prairie on a nice warm summer day that I'm very much missing with this Omaha weather. So at the prairie, I would capture monarchs, and then I would take this gel substance here in a vial, and I would swab the monarch on their feet and the proboscis. I'll go a little bit more in depth with this with a video, but just to kind of give you some background, that is what I'm doing at the prairie. And then I'm also analyzing these samples in lab. This is the step I'm currently on with my research. So basically how I analyze them is I melt down the gel on a microscope slide, and then I look under a microscope to find the different pollen grains. Also with the collection step, I did a prairie survey of all of the flowers in the prairie. That way, once I found pollen on the monarch slides, I could identify it with the species from the prairie survey, and I analyzed the samples in the similar way. So kind of going more in depth with my methods, I made a video over the summer just to kind of explain things because it is a little bit more complicated.
Hello everyone, and welcome to the methods section where I am at the Glacier Creek Prairie Preserve. I have already taken the liberty of catching a monarch, so you don't have to watch me goofily waving around the net. So I will go ahead and grab him or her. So here we have the adult monarch, which I will be swabbing for pollen. So what I like to do first is I like to sex the insect, which this one is a male monarch butterfly. And these are actually a little bit more rare to catch because male behavior tends to be flying around a lot more in the prairie and not landing on flowers. So it's pretty awesome that I caught a male right off the bat. So first I take my prepared gel slide and I swab the feet of the butterfly. So as you can see, there's tiny little feet on him, and we swab the feet and the kind of the leg areas because there's tiny little hairs that can catch lots of pollen. So this is kind of one of the primary areas that if pollen is going to be on the monarch, it is going to be here. To make sure that I label this right, I literally put it at my feet. And then next, I grab another slide, and then what I do is we are going to be swabbing the proboscis. And the proboscis is basically kind of a long straw on the face of the monarch that helps get the nectar. So I have a little sewing pin here and I very carefully go and unroll the proboscis. So that's the proboscis there, very long straw. It's what the monarch uses to get nectar out of the flower, so it could be another area where there's pollen on the proboscis. It's important that we swab 2 different areas so that we can kind of tell where the monarch is holding the pollen. It does not want me to do this. In an instance where he's not letting me do it, I can swab the needle quite a few times just to see if I can get any pollen, or I can kind of manually unroll it with the end here. There. And then we say goodbye. There we go. Alrighty, thank you for watching.
Yep, so that's a fun little video. So here is the data I have so far. This is my lab analysis portion. So this is the pollen from the butterfly gels. These are 2 different butterflies that I was able to catch, and both of these pollen grains were found on the feet of the butterfly. So one of my hypotheses is true, that it's likely like the hairs on the legs and feet of the butterfly that allow pollen to attach to it. And these are 2 species of flowers that I was able to get pollen grains from for my prairie survey. So the one on the left here is compass plant. This is the sunflower variety of flowers. And as you can see, the pollen here is very, very spiky, and it looks like it would easily attach to something. And the one over here is wild bergamot, and it's a little bit smoother, and it has a pretty unique shape for pollen grains that I've seen before. And the compass plant and aster sunflower variety of flowers is more likely to be found on the butterfly samples, just because it will stick to either the legs or the proboscis a little bit easier. And generally, I see more monarchs on these types of flowers than on the wild bergamot in species number and in just general placement of the flowers in the prairie. So what I have left to do for the Monarch project in 2021, I need to finish examining the butterfly gel slides. Each gel slide takes quite a bit of time to analyze, probably about 2 to 4 hours per slide, and each butterfly has multiple slides, one for the feet and one for the proboscis, so it does take quite a bit of time to get one butterfly completely analyzed. And then I need to compare the pollen samples from the butterflies to the flower samples. That way I can get a good good knowledge of the species that I'm finding that the butterflies are picking up. And then I'm going to compile the data to show trends. So far, the butterflies are carrying the pollen on their feet, but that doesn't mean that they're not able to carry it on the proboscis. It just means that I wasn't able to swab successfully the pollen. That could still be possible. And then also, like I said before, to know the species of the flowers that the monarchs are more likely to pollinate. So the future research impact. So further research should definitely be on examining the flower visits of monarchs, and my data could be used to kind of target different species of flowers that monarchs may be able to successfully pollinate based on like what types of species are more prevalent in my study. And then another study could look at flower germination, again, of these flower species that I am able to collect and what the monarchs are mostly visiting. One could collect the flowers and see if germination is occurring from the butterflies. And then environmental protection. Most of this is for the lack of monarchs that we have right now, the population is decreasing rapidly, and unless we get it to a more sustainable population size, we could easily see monarchs to keep declining as time continues. And then this will also generate public interest. The more we study monarchs, more people will hear about it, get involved, and make more conscious decisions about their environmental impact. So monarchs could be a great way for people to kind of get into environmentalism and be more conscious of their conservation efforts. And hopefully that would also be beneficial for the butterflies and increase their population. So like I said, here we have a graphic from Monarch Watch. The most updated year is 2.83 hectares of butterflies over the summer. And you can see it's a rapid decrease from the past years. And unless we get the butterflies to a more sustainable average, they could keep declining. So I want to say a big thank you for everyone listening. Feel free to come to my Zoom room for questions. And if you can't make it that time, here is my Creighton email. Please email me any questions. I also have tons and tons of sources and photos if you have any general interest or questions for me. A big thank you to Dr. Taylor and Dr. Burke. They were incredible mentors during the pandemic. And I would also like to thank CURAS Surf for funding for this project. And also a big thank you to every staff member that I don't see that has a big impact of keeping research available during the pandemic. Thank you very much.